Is the another coming or not coming? Hi. So everyone, good evening again. Hope everyone can hear me. Can you all hear me? Hi. Last day we started discussing about water potential concept. I'll be asking few questions from you guys. Just wait. Right. Um, what are the factors affecting for water potential? What are the two factors, let's say? Number one is that. So please start writing along with me. So you can write the sorry, I got disconnected. Can you all see the board? Number two, write the water potential equation. Number three, a cell has a water potential of 0.3 megapascals. It has a pressure potential of, this one has to be minus, of 0.7 megapascals. Find the solute potential of the given cell. Do these three questions quickly, all of you. හරිද පුතේ ඉවරද? ඉවරද? Hi. First question I'll be discussing. What are the two factors affecting the water potential? What do you think? What are the two factors?
Solute potential and pressure potential.
Yes, solute concentration gradient, isn't it? Am I correct? And number two? Where is it? What is number two? Physical pressure applied, isn't it? Did I answer correctly, Pute? Hi. Write the water potential equation. Psi is equals to psi s plus psi p. So you have to write it and define the term. Psi is equals to psi s plus psi p. What is psi? Psi is water potential. Then what is psi s? Apa tu?
Solute potential.
Solute potential. And what is psi p?
Pressure potential.
Pressure potential. Yes. Further. Okay, everyone. Right. Now there's a question. It says that the cell is having water potential of minus 3 megapascal. So we have to write the equation always. Psi is equals to psi s plus psi p. So the given values we are going to apply. Minus 0.3 is equals to 0. No, this is not given. No, psi s plus 0.7. So you are getting value of psi is equals to minus 1.0. Am I correct? What's the unit here? Megapascal. Another.
Yes, yes.
That day I discussed about flaccidity, turgidity, and those things also, no? Isn't it? Right. Wait a second. I'll explain again with another diagram. Right. You can see the diagram now. Now think if you are putting this cell in a solution which is having less water potential than the cell. So what's going to happen here is water is going to come out of this cell to the solution. Am I correct? The water will move out. Until what time, Pute? Water will move out until an osmotic equilibrium is reached. Until an osmotic equilibrium is reached, water will come out. What will happen exactly at the osmotic equilibrium is, if you think that two water molecules are going out, two water molecules should be taken in. That means both directions the water flow occurs. It does not stop, but still no net movement, right? That's the correct term to describe here. No net movement of water at the osmotic equilibrium. Do you agree with that?
Yes, teacher.
Do you agree?
Yes, teacher.
Hi. So now this, because of movement of water to outside, there is a rigid outer covering which is the cell wall. The plasma membrane is tend to detach from the cell wall, isn't it?
Yes, teacher.
Plasma membrane cell wall again detach when, all right? So that incident we call it as plasmolysis. The cell is now known as a plasmolyzed cell. This can be different stages. Give me 1 second. Look at this picture I have placed here. It starts from a turgid cell which is having cytoplasm is fully filled with water, which is giving some pressure towards the plasma membrane. So this number A is a turgid cell. So when the water starts to move out of the cell, there is one point the cell membrane detaches from the cell wall. This is the first initial stage. So this is the stage that we call it as incipient plasmolysis. At the incipient plasmolysis, you know what? The psi p of the cell is now becoming 0. Let's say initially this cell had a psi p value of, let's say, 0.7. With the removal of water, psi p gradually reduces, and finally, as soon as the cell membrane detaches from the cell wall, it is known to have psi p 0. Do you all agree?
Yes, teacher.
The first movement, the psi p became 0. We call it as incipient plasmolysis. Remember very well, thereafter, in the cell, psi p is always 0. Psi p is 0 in diagram C. Psi p is 0 in diagram D as well. So with the time, with the more movement of water towards the solution, the cell is more and more detached. That means the cell contents are more detached from the cell wall, and finally it will be coming and it will be going to a corner of the cell and staying like a small portion. So what is exactly filled in this area which is seen empty? What is coming to this area? Can anyone guess? What comes here? External solution. This is filled with the internal solution. Do you agree with that? That
Yes,
means
please.
if it is a sucrose solution, it is the sucrose solution filled here. Got it?
Yes, please.
Hi. Now everyone draw these four cells. It doesn't have to be exactly this one. You can usually, we are usually drawing with two lines on the cell wall. Can you draw a few cells, four cells, and show what has happened as stages and mention that some psi p values I have mentioned here, that one, as well as this number be diagram as incipient plasmolysis. All others are not incipient plasmolysis, but they are plasmolyzed. All B, C, D stages are plasmolyzed. Understood? Got it? Draw them quickly. Thereafter, we will be going to one practical from this. Please draw. Finished drawing?
Yes.
Last one.
Okay. All
Done.
done? Hi. I'm erasing all. Hi. Now I am going to the practical handbook. We will be discussing one practical now. Wait. Right. So we will be discussing this practical today. Determination of solute potential of epidermal fields of Rhoeo or Tradescantia. So that is the scientific name here. Write down the topic, everyone. Rhoeo is the scientific name. You must underline that one. How does it say? Done writing? Finish?
Yes, teacher.
Are you done? Go. Tika.
Yes, teacher.
Right. So, we are going to discuss this one. What we are finding here is, we are finding out the solute potential of the Rhoeo lower epidermal fields, right? So have you all seen Rhoeo? First of all, tell me that one. Rhoeo dekha tinada? You may have this in your home garden also. This is the correct plant here. Visually, we are using for our practical, right? So, have you seen this one?
Yes, teacher.
Hmm?
Yes, teacher.
Ah, 1 minute. Right. So there is a particular reason why we are using Rhoeo in this experiment. It's because of the color of this lower epidermal pale cells, so that it is easy to observe. There is a vacuolar pigment in the Rhoeo leaf that is anthocyanins, which are found in the epidermal cells, lower epidermal cells, especially in the Rhoeo plant. Hi. We'll go with the questions also so that it is easy for you to continue also. Please write down the question. Number 1. What is the reason for using Rhoeo in this experiment? What's the reason for this? Because it is easy to observe the epidermal cells, especially the plasmolized cells, due to the vacuolar pigment anthocyanin present in Rhoeo lower epidermal pale cells. Write down the answer. You have to underline the scientific names. Done? All right. So, first of all, in this experiment, you all have to prepare a solution of sugar. So you are using normal brown sugar, which is in the market, and then you are preparing 1 m molarity 1 sucrose solution first, and then using that one, you are going to prepare a concentration series, right? So here, these are the concentrations that we will be preparing: 0.15, 0.20, 0.25, 0.30, 0.35, and 0.40 m solutions are produced, right? First of all, before you peel off the Rhoeo, you have to, it is a must to prepare the concentration series, because if you just peel Rhoeo leaf beforehand, it will dry and you will not get the proper result, right? So as soon as when you take the peels, you have to put it into the relevant solution and close the lids of the Petri dishes, right? So it is taking a concentration series and you are adding the solutions to the labeled not actually test tubes. You'll be using Petri dishes, right? Using test tubes, you may be pouring it into Petri dishes, but anyhow, you will be preparing 6 Petri dishes which are having the relevant solution poured. And then you are taking Rhoeo lower epidermal peels. How do you take this one? Think that this my paper is Rhoeo leaf. You have to tear it. Okay, give me 1 minute. Just think that this is my Rhoeo leaf. You have to take the lower side to this side and you have to tear it quickly. When you tear it quickly, you will get small thin peels which are at the edge of the leaf. Then you are using a needle or a forceps, or and as well. Taking tiny pieces and you'll be putting few pieces to each Petri dish. Understood up to now? Do you understand up to now?
Yes, teacher.
Hi. Now we have 6 Petri dishes here. I think these are the Petri dishes. 1, 2, 3, 4, 5, 6 are there. So once if you put a few pieces into each of the solution, what you're going to do is you are going to keep few pieces and you are going to close the lid also. Now my question is, why you are closing the lid of the Petri dish? Write down that question, everyone. What is the reason for closing the lid of the Petri dish? Right. My second question was, what is the reason for closing the lids of the Petri dishes? Can anyone know? Can anyone tell what's the reason for closing?
To prevent air getting inside.
No, that's you're closer, but it's not the answer.
To maintain constant pressure potential.
No. The reason is this: if the epidermal peels are coming to an equilibrium with the environment, our experiment goes wrong. Right? Ensure that epidermal peels are coming to an equilibrium with the given solution. Right? Write the answer there. This is to ensure the epidermal peels not coming to an equilibrium with the environment, the air or the environment. Right? Okay. Harga.
Right.
Okay. Right. Now we'll be moving to the next part. Now you are going to keep it for some time. How long we are going to keep it to come an osmotic equilibrium? Can anyone tell me? කොච්චර time duration එකක් තියෙනවද? How long will it take for the epidermal field to come to an equilibrium? කොච්චර time එකක් දෙනවද? අපි 20 minutes time එකක් දෙනවා, right? Write this down. 20 minutes. හරිද පුතේ?
Yes, miss.
So my next question, after 20 minutes, what will happen to the epidermal peel? What do you think? After 20 minutes, what will happen, Pute? The epidermal peel will come to an osmotic equilibrium with what? With what? The relevant sucrose solution. Agree? At this stage, as we discussed before, equal amount of water goes in, equal amount of water molecules goes out. Right? That is the scenario. Hi, write down the correct answer there. Answer the question both. Adbute, finish?
Yes, miss.
Right. So then what we are going to do after 20 minutes time. Let's come to the here. We have done this place, right? Just take a little time and read up to this point.
මිස් මේ නිසර මාකර දෙන පල්ලෙහා ටිකක් කියවන්න ඕනේ.
නෑ මෙතෙන් වෙනකන් කියවම ඇති. දැන් ඔස්මොටික් ඉක්විලිබ්‍රියම් එකක් හැදෙනකම් විතරයි අපි කිව්වේ මෙච්චර වෙලා. ඒ ටික කියවන්න පුළුවන් මොකද්ද කරේ කියලා. ඔතෙන් වෙනකන් කියවන්න. මම මෙතනින් එහාට තාම කියලා දුන්නේ නෑ. ඕකේද? Hi, Bhuti. I'm going to next part. Are you done? Then what we are going to do is you are going to use a paintbrush. You know, now the Petri dish is here. Using a paintbrush, you will be taking a thin epidermal peel, right? So think this is the brush. You are taking a thin epidermal peel. How would you find out a thin epidermal peel? Now this is in water. Now you can see, if you can see through, it means it is very thin. What's the disadvantage of having thick epidermal peels here? Let's write it as a question. Why are we selecting epidermal peels? And how are they selected? What is the disadvantage of selecting thick epidermal peel? Write down the question. Right. Why are we selecting a thin epidermal peel? As it's thin, there is no overlapping cell layers, so that it is easy to observe the plasmalized cell. Right there, it is easy to observe plasmalized cells when without any overlapping cell layer. Okay. So the opposite of this coming as the next answer, what is the disadvantage of selecting a thick epidermal peel? What is the disadvantage? Overlapping layers will disturb the agree? Okay then.
Okay, Miss.
Now, what you're going to do here is you will be taking the epidermal peel from the from one sucrose solution, and you are going to take a clean glass slide and you are going to place what is in the middle. Can anyone tell me what are we going to put in the middle here?
Sucrose solution.
Yes, you have to definitely take the relevant sucrose solution and put a drop here. Do not write you'll be putting water droplet. That's very wrong, okay? So you'll be putting a relevant solutions droplet, and then you'll be keeping a thin epidermal peel selected over here, and then you'll be closing it with a cover slip, and you'll be removing the excess water. After that, okay?
Teacher, can you repeat it one more time?
Yeah, okay, sure. You are taking a clean glass slide and you are taking a droplet of relevant sucrose solution, let's say 0.25. You'll be taking a drop of relevant sucrose solution and placing it on the slide, and you'll be selecting epidermal peel from the same solution, and you'll be keeping it on top of the water droplet, not water, the sucrose solution droplet, and then you'll be closing it with a cover slip. Up to that, okay?
Okay, teacher.
Then you are going to remove the excess water using a blotting paper. Now I'm going to the practical book again. Instruct them to set the Petri dishes aside. That one I discussed. And yeah. Direct the students to mount the fragments of each epidermal peels on the slides using paint brushes in a drop of sucrose solution from which peels were immersed. Okay. Relevant solution they have highlighted. I'm asking the next question. What reason for placing a droplet relevant sucrose solution on the last slide. Why not a water droplet? Now write the answer. You have to write this answer. Hello, did you write the answer? What is the correct answer there? What is the reason for using droplet of the relevant solution.
Instead of becoming plasmolized, the cell
हाँ, ये बताइए।
will become turgid.
Louder. What?
Instead of the cell becoming plasmolized, the cell will become turgid.
Yeah, if you put water, surely it will become turgid because it's coming into an equilibrium with water. That is the answer there. But why are we putting water? Otherwise, we could have put without anything, no? Hmm? Why is that? Otherwise, the epidermal peel will dry before you observe, right? To make sure that to avoid drying the epidermal peel. And if water is added, what will happen? As the epidermal peel will come to an equilibrium with water, and it will affect the results. Do you agree? That's good thing. All right. Now, what we are doing is we are preparing the slide and we are going to mount it in the microscope. Can anyone tell me what's the procedure that we are doing? We have discussed this in the first practicals now. What is the procedure? Where you are keeping the slide?
On the stage,
On the stage. Then how you secure it?
using stage clips.
Hi. Then you will be turning which power lens here?
First, the low power lens.
Low power lens, and then what you should do?
We should adjust the coarse adjustment.
No. Before that?
Focus the light.
Yeah, you have to adjust the light and you have to make sure that enough light is coming through the objective lens. And then you will be focusing using the which focus?
Coarse adjustment.
Yes, coarse adjustment or rough focusing knob. And then you're taking a clear field and then you have to turn it into the mid power here according to the practical. Mid power, you are using which adjustment knob?
Fine adjustment.
Fine adjustment knob. And then you will be making it more clear. Right now I'm going to show you a few pictures that can be seen here. You can observe pictures like this. So what we are looking here is we are going to find out the plasmolized cells here, right? Wait. You can see it properly now. So what cells we can say it as plasmolized cells here? The cell membrane has to detach from the cell wall. Can you see that this cell is plasmolized? Am I correct?
Yes, teacher.
But some cells are not plasmolized. Here, in this case, many cells are plasmolized, but some cells like this, you can see that it's not plasmolized. It's kind of a filled, no? Isn't it? So those cells are considered as turgid cells. What we are going to take here is you are going to count the total number of cells here. Total number of cells you are going to and you are going to count number of plasmonized cells also here. Then you have to multiply this by 100. So what are we going to take it here? We are going to take the percentage plasmolysis. Percentage plasmolysis is equals to number of plasmolyzed cells divided by total number of cells in the microscopic field and multiplied by 100. Okay, got it? Then on.
Miss පොඩෝක clear මේ slide එක පොඩ ලියලා දෙන්න පුළුවන්ද? මේ
Alright, sure. Wait. Hi. Percentage plasmolysis is equals to number of plasmolyzed cells divided by total number of cells. You have to multiply this by 100. Understood now?
Yes, teacher.
yes please.
The total thing has to multiply it by 100. So now you have how many sucrose solutions here? There are 6 solutions given below. So you have to understand that you have to take this for all 6 solutions. Understood? Got it?
Yes, teacher.
Hi. Now I'm going to the practical book again. Hi. Now read these sentences up to this. Can you see it clearly? Today I'll be finishing at 9 o'clock. Next day I'll give more time. Had the Buddha finished reading, everyone? Hi, then what we are going to do is you have to take the percentage plasmolysis for all 6 solutions. Did you all think why did we do this one? The reason why we did this one? Did you all having any thought why we have done this? Do you have any idea? Molarity of sucrose solution versus percentage plasmolysis. So the given values were we had 0.15, 0.20, 0.25, 0.30, 0.35, 0.40 should come here. Alright, then you have to write the percentage plasmolysis here. So after this, we are going to draw a table. Can you guys tell me what comes to x-axis and what comes to y-axis of the table? Here it is y-axis. Here it is x-axis. Can you guys tell me what comes to x-axis and what comes to y-axis?
Y-axis is the molarity of the solution and x is plasmolysis percentage.
No, x-axis is the molarity of the sucrose solution. That is the independent variable. What we are changing here is here, right? Molarity of sucrose solution comes here. Others agree? And to this side, I'm taking percentage plasmolysis. Are the 1 second, please. Hi, can you all find out a graph paper, or you can use a square ruled page also, else you can use the usual normal sheet and you can do this one, okay? Right, I'm going to give you a few data. Wait a second. Hi, I'll add it to the table itself, so you can start drawing the graph. I want everyone to draw the graph and show it to me, and make sure you will spread it and draw. Don't draw tiny half of the sheet graph. Full space should be utilized properly. Okay. 0.15, 18%. Right, start drawing. And zero point 0.20, 32%. And 0.25, 48%. Then 0.30, 68%. 0.35, 80%. 0.40, 90%. Alright, quickly draw the graph before the class ends and show it to me, everyone. Okay, I'm waiting here. විමේශක graph එකේ ෂෙනාගේ graph එක හරි විමේශක බිඳුවට attach කරන්න. අනේ මේ කවුද යෙනොල නම් graph එක වැරදිනේ පුතේ. graph paper එකක් කරන් ලේෂල්ලි යෙනොලගේ එක අයි උනේ මොකද දන්නවද? ඔයාගේ උඩ axis එක spread කරලා 1 minute please. තව කවුද එව්වේ නැසිකෝ මේ දසිකෝ මම පුතේ මෙතනින් ඉහාට කන්ටිනියු කරන්නම්. කියපු ටික තේරුණානේ මොකද්ද කරන්න කියලා? ඊට graph එකේ මොනවද ගන්න කියලා ඒක ඔක්කොම ඊළඟ දවසේ කියලා. දසිත් මට graph එක එවලා තියන්න. ඕකේ?